Chapter 5 Membrane Transport and Cell Signaling by Jessica Peck and Oliver Knöcklein. Saturated versus unsaturated fatty acids. An unsaturated fatty acid is liquid at room temperatures, healthy, and has a bent tail. A saturated fatty acid is solid at room temperature, unhealthy, and has an artificially straightened tail. Selective permeability. The fluid mosaic model explains how membranes regulate cell traffic. It is a lipid bilayer which is nonpolar and hydrophobic. Nonpolar and smaller molecules can easily pass through. Types of proteins. Integral proteins penetrate the hydrophobic interior. They are hydrophobic and they are in the transmembrane. Peripheral proteins are not embedded within the lipid bilayer. They are loosely bound to surface for structural support. Transport proteins span the membrane, are hydrophilic, so polar molecules can pass through. Channel proteins are tunnels for certain molecules. Aquaporins are types of channel proteins which help water pass through membranes. Types of transport. Passive transport is diffusion down the concentration gradient. Osmosis is the diffusion of free water across a selectively permeable membrane. Facilitated diffusion is diffusion down concentration gradient with the help of carrier proteins, which serve as gated channels. Active transport is diffusion against the concentration gradient, which requires energy. Hypertonic, hypertonic, and isotonic solutions are different levels of tonicity. In an isotonic solution, there is no net movement. In a hypotonic solution, there is net movement into the cell, and in an hypertonic solution, there is net movement out of the cell. The sodium-potassium pump is a type of active transport which uses membrane proteins. The pump moves 3 sodium out of the cell and 2 potassium into the cell to create an electrochemical gradient. Exocytosis is when a cell secretes certain biological molecules by fusion of vesicles with plasma membrane. Endocytosis is how a cell takes in molecules and particulate matter by forming new vesicles from plasma membranes, it is the reverse of exocytosis. Phagocytosis is the cell eating, penocytosis is the cell drinking, and both these methods help rejuvenate plasma membrane. The diffusion and osmosis lab. Part 1. The experimenter places a dialysis tubing bag with glucose and starch on the interior in a beaker full of distilled water. After 30 minutes, both the interior and exterior are tested for glucose and starch using glucose test strips and IKI solution respectively. The results will demonstrate the movement of different molecules against the membrane. Part 2. The experimenter places dialysis tubing full of 20 milliliters of different molar solutions in distilled water after massing them. After 20 minutes, they weigh the tubes again, recording the amount and observing differences between the initial and final mass, evaluating whether they are isotonic, hypertonic, or hypertonic. I hope this has been helpful.